Welcome back. We are now moving on to uh, protein sequencing and other kinds of technologies that can be classified as high throughput. And any word that you see in biology that has the word omics, this slide talks about omics technology or massively parallel. So what that means is one can do these top-down techniques at one time. So you can have all these different technologies happening at one time. So we're going to be talking a little bit about mass spec. We've talked a little bit about that. We certainly have talked about 2D gels. Probably in molecular, we'll hit sequencing and microarrays. But I just wanted you to realize there's a bigger picture out here, and a lot of these technologies give us broad top level work here. It allows us to find new pathways, and then we can model these pathways together in a cell. And that's called systems biology. So systems biology takes the pathway information, tries to link it together. Eventually, we're talking a synthetic cell. I mean, that's where, so the synthetic biologists, they're here now. It's just the technology hasn't, uh, you know, got up with the synthetic biologists. But eventually, we will have enough model to put the right pathways into cells and experiment to alter function or do activities we probably haven't even thought of yet, which is good. I mean, usually it's hard to predict what technologies will do. But omics, if you see the word omics, that's usually a high throughput, big data kind of approach to biology here. And I thought this was, I really wish we'd had this slide a little earlier, but I found it tonight. And it kind of starts where we had stopped. This is protein extraction here. Here's from the culture media. Uh, Discusses different ways to get cells here and just talked about finding the right proteins, having the right chemistry to find those proteins. We can take those proteins and separate them by chromatography, different ion exchange, uh, size exclusion. A lot of the technology we've talked about, we hadn't talked about boronic acid or whatnot, but in lectin affinity. Just since we're here, lectins are molecules that bind sugars. So lectins actually can bind the sugar trees on glycoproteins. So you could have an affinity column that had a certain lectin that would bind certain mannose or NAG-rich or galactose-rich proteins, if that was an interest. And just to let you know that in cancer cells, sometimes the post-translational modifications, and those you might see PTMs. The PTMs, okay, of proteins can be different based on diseases, and that could be definitely a biomarker. So there's a way you can actually purify specific types of glycoproteins called lectin affinity chromatography. And we can separate proteins by 1 or 2D gels. And so we've talked about that. And we had mentioned that sometimes proteins are so big that you can't get information about the whole protein. You have to break it up. And there's an enzyme called trypsin, and a digestion with that protease. It's a protease. That means it chops up proteins. It's called a tryptic digest. And those then small fragments can be analyzed with mass spec. And we've talked about liquid, the electron spray mass spec here, the charge to mass ratios. We're going to cover Maltitolf, time of flight, mass spec. Okay, you can imagine a big molecule has probably going to be a little slower than small molecules, so the time of flight would be different. And we're going to talk about Edman protein or amino acid sequencing. Dr. Edman invented a technique where we can take one amino acid at a time and determine what it is. And it starts from the N-terminus. So go back to your protein structure and realize that their proteins have an N-terminus, a C-terminus, and the different amino acids are held by peptide bonds. Well, Edman degradation clips those bonds and takes the amino acid and analyzes it, what came off, kind of like pearls on a string, and you can clip each pearl off and study it. And then eventually we're going to take this high throughput omics data here and go through our bioinformatics. We can identify proteins, we can predict localization. Certainly in cell biology, we're very interested in where in the cell. We've certainly, with our inquiries, understand functional domains here. And here are these post-translational modifications that could include carbohydrates, could include lipids, other kinds of modifications, and processing occurs after the proteins are made. Post-translational, after proteins are made, they can still be modified. And so sometimes I can determine a difference between a protein's function in one type of cell, say a liver versus a kidney, is the modifications might be different. But anyway, this is kind of a big picture. Again, I like big pictures here. And so looking at proteins, the word is omics, proteomics, proteins, big data here, high throughput, means you can do a lot of samples at one time, a lot of patients at one time, a lot of samples at one time, and look for biomarkers. And so we can look in each sample at the protein components. We get a snapshot, and two-dimensional gels can be a snapshot. Here the 2D is. We can compare tissues, cells, certainly diseases, drug treatments, all sorts of different aspects we can compare and get a differential look at the type of proteins that are being produced under those conditions, cells, tissues. In molecular, we'll talk about the type of messenger RNAs that can be made under these conditions. And so the proteins made under disease states, that gets back to what proteins are there, what proteins are up or down. We can find proteins that could be a drug target. Maybe we want to shut the protein down. Maybe the protein's not supposed to be there. Well, let's quiet it down, shut it down. Or we might be able to find a protein that's always found when there's, say, cancer. It's called a biomarker or a diagnostic marker. And you can automate these by robots or semi-automatic procedures here. And we can get a lot of data at one time using the robots. So it's high throughput here. So when we look at proteomics, just realize that there are ways to look at proteins in a structural way. We're going to talk maybe a little bit about protein crystallization. There's how you can model proteins. We won't hit nuclear magnetic resonance spectroscopy, but you can model in a three-dimensional way where amino acids are. But anyway, you can compare a mutant versus a wild type protein. How about a mutant RAS versus a wild type RAS. You can look at crystallography of those two proteins and see what's different. Expression or analytical means that we're looking at the amount of protein under these conditions. We can do the 2D, 1D electrophoresis. There are things called protein and DNA chips. We'll talk about that. HPLCs, mass spec, and sequencing proteins by mass spec or Edman degradation. It's called micro sequencing. Basically, they're not want to sequence the whole protein, but you want to sequence enough to figure out what the protein is. And then you can measure the amount of protein. That's called expressional or analytic. And then finally, the third flavor of proteomics is functional or interactive. We will cover yeast 2 hybrid, and you guys have been doing motifs and domains here, okay, in your inquiries, but we're looking for function, and we're trying to figure out what we can detect there. So three flavors of proteomics. This is just, I found this on the internet. Just that there are labs now that you can send samples to, and they will apply all these technologies and give you information about those samples. So in the bigger research places, anybody that's wanting to do a lot of high throughput work, you need near these labs, okay, and a lot of big John Hopkins, for instance, other, Harvard, the medical school. These places have these just right down the hall. You just walk in, here's my sample, tell me everything that's in there and how much. And you might say, compare it to this sample. So here's a cancer cells, wild type cells, tell me the difference. And that kind of data is really interesting, allows doctors, again, to predict diseases and treat diseases here. Sometimes the data can get even more in depth, and so we've talked a lot about how we identify proteins, and we'll talk a little bit more about sequencing and mass spec. Here's our modifications, okay, of different kinds. Modification could be, is the protein phosphorylated? Does it have a sugar tree? It's glycosylated, methylated, acetylated, ubiquitinated, all sorts of different modifications. We can figure out the protein structure. How much protein is there? Okay. And is there a metabolite? In other words, Dr. Grace, if you're familiar with his, he does metabolite work. In other words, these are the signature products of the different pathways. And these are molecules and chemicals that are the result from pathway activity. And certainly you can figure out structure and mass spec determination of molecules or compounds there. So the big picture, and this is just hardening to arteriosclerosis. Here's a healthy artery because of genetics or environment, diet. It's got a plaque here. And so now we can study cell cultures, animals, or clinical studies and apply our omics. There's all our omics. Okay, we're in cell biology, we hit proteomics, but there are others here. And of course, you can look at the databases. Okay, and those data, and certainly in the literature, you can find what's been done. So all these different omics, literature, and any databases come together, and you're trying to model exactly what's going on here. Why are we getting plaques? How do we reverse it? So that's the big picture. I mean, from the clinical side, all these techniques allow us to apply this information to model a disease or a situation. If we can model it, we can break it down, we can maybe cure the disease. So we had talked about uh, electrospray, and we will talk about Maltitol time of flight. And again, this is protein sequencing. We need to understand the sequence of the protein we're studying. Amino acids have R groups. They impart uh, locational information and functional. And again, there are post-translational modifications that the mass spec can determine. Edman cannot. So Edman cannot do post-translational PTMs, mass spec can. And of course, if you find the gene or clone the gene or have the gene, then you could figure out the sequence that way. So not every protein has to be sequenced. The only thing, though, is the modifications are not going to be found in the sequence. So that means that a lot of times mass spec, the proteomics, imparts more information than you would predict because there are different modifications that can occur. So informatically, you can predict the amino acid sequence if you have the gene, but it may not tell you exactly what the modifications are. So this is Edman degradation, developed by Dr. Edman. It's the classical method. It's sequencing amino acids in a peptide. And there's a series of chemical reactions that occur, and I think they're listed here. And fortunately for this situation, these are cyclic, and these reactions occur on one amino acid at a time. So that's why this is able to chew down a protein sequence doing the chemistry one at a time. So in this method, the amino terminal residue, the N-terminus, is labeled and cleaved from the peptide, and it doesn't chew up or break the other bonds. So in the first reaction, amino acid 1 is released. Then the second cycle, amino acid 2, 3, 4, etc. So the chemistry occurs. Here is this compound. The Edman reagent is called phenylisothiocyanate, PTC. There it is. That's PTC right there. This molecule. And you see this interaction here. This was a nucleophilic attack. I used to know this stuff. Anyway, what happens is a bond is going to form here under basic conditions, pH 8. And so what happens, here's the peptide, here's the first amino acid, there's its R group, changing the pH to more acidic, and you heat, that causes the bonding to occur. And notice that the sulfur group, we have a circularization, and eventually the peptide, so amino acid 2, and all the other amino acids are released, and the product, the intermediate product, is here with a little more acid and heat. It becomes the final product. It's phenylthiohydantin, whatever amino acid it is. So PTH and amino acid. So we have 20 amino acids. Okay. So every amino acid is different. And so the Edman reaction, going through the PTC, changing the chemistry here to eventually release the first amino acid here, and then you form the PTH adduct with the amino acid. Each one will have a different amino acid PTH compound. That's a key point. Edman degradation doesn't work unless you get one of 20 in the product. So this is called the cycle, right? And so now this peptide with amino acid 2, okay, is applied to the PTC reaction, and you get it off, and you'll have a peptide with amino acid 3 starting. And this will go 20 to 50, depends on the efficiency. You can get 20 to 50 amino acids sequence. If protein is bigger, you have to chop it up. You have to trypsinate it. So it's a cyclic chemistry. There's the PTC coming on, high pH. Here's the peptide. Here's the phenylthiocarbamide derivative. Here's some more chemical names, anil, thiazonal, whatever. Eventually, and here's the peptide shortened by 1. And then there's the PTH, phenylthiohydantin derivative of whatever amino acid. And that's shown in purple. And again, one amino acid at a time is released. And here's the concept again in a diagrammic form. First amino acid is off. And so how do we figure out what it is? HPLC. Every amino acid is different, and therefore it will have a different signature on the HPLC. You're saying, well, how do you know what's what? Well, obviously, they've done it now enough to know exactly what the profile is of each amino acid. And the machines are calibrated such that we can determine each amino acid. And here's the next cycle with amino acid 2, would start right back over again right here. This will be added, and you take off number 2. Here it is. And it's analyzed by HPLC. You can analyze 20 to 50, so probably average around 30. It's very reliable. And if the protein's big, you'll have to cut it into fragments. And each fragment then can be sequenced. And this is just showing how we do mass spec analysis. And we've talked a little bit about that. So here's the absorbance at 269 nanometers. And these are the fractions that came off through the Edman chemistry. And you can see they're 1-letter amino acids and number of fractions. So this is, I can't see the first one, but here we go. Cycle 1, you get alanine. Cycle 2, glycine. Cycle 3, leucine. Cycle 4, proline. Cycle 5, valine. So you actually get a profile, and the computer then records said profile. And you're saying, what happens? You get contamination. Then you get multiple profiles, and you could not figure out which amino acid went to which protein. So Edman requires complete purification. So if you did a 1D gel, cut a band out, and you tried to sequence it with Edman, most likely you've got contamination. 2D gels allow us to have a better shot at getting a pure protein for Edman degradation. So this chemistry occurs until everything, all the chemistry and everything so degraded that you don't get a good profile. Notice the absorbance and the levels. Over time, the levels drop. I don't know if you can tell. Look at the peaks here. At some point, they start dropping, and you start getting, there's a threshold where you can't read anymore. There's a threshold. So they've taken Edman, and in this case, they've actually put it to a solid surface and kind of automated it that way. And here's the phenolthiohydantin amino acid, and then they identified each cycle there. And then once you get the amino acids, you can search the database. What protein is this? So Edman versus mass spec, protein identification using peptide sequencing. Here's the peptide. It takes a while. The chemistry is hours, okay, 12 hours. Mass spec looks at all profiles, all different fragments of this region at one time, and you get 1 second and you get all the data, or 12 hours. So that's part of why Edman degradation is in fact classical versus the mass spec analysis, which is the newer way of doing that. And it's just showing how they're going from amino acid page and fractions all the way to sequencing in here. It is showing that. Okay, so I'm going to finish here with the Edman. We will pick it up with 2D gels of the lysate, and we'll continue into the mass spec. Come back.